Hello everyone, welcome to the study of points about Section 2, Epigenetic Regulation. The different types of regulation involved in epigenetic includes DNA methylation, histone modification, non-coding RNAs, and chromatin remodeling. Understanding the roles of epigenetic regulation in DNA replication, DNA repair, transcriptional regulation, and chromatin package can give a deep insight in cells' development and the causes of the relative diseases. The principal type of epigenetic modification that is understood is DNA methylation. DNA methylation is a covalent modification of genomic DNA that modifies its gene expression and provides mechanisms for transmitting and perpetuating epigenetic information through DNA replication and cell division. The role of DNA methylation in cell regulation has also provided the potential for a new paradigm of disease intervention and treatment. Under normal dietary conditions, absorbed folate is metabolized to 5-methyl tetrahydrofolate. The abbreviation is 5-methyl-THF in the intestine and liver. This reaction is key to maintain the flux of methyl groups for the remethylation of homocysteine to methionine via the vitamin B12-dependent methionine synthesis reaction. 5-methyl-THF is then back to THF for the next folate cycle. Methionine is a substrate for SAM, a cofactor and methyl group donor for numerous methylation reactions, including the methylation of DNA, which is involved in methionine cycle. Under the catalysis of DNA methyltransferase, the abbreviation is DNMT. It is used the methyl group provided by SAM to methylate the 5th carbon atom of cytosine, thereby converting cytosine into 5-methyl cytosine. The abbreviation is 5-MC. This covalent modification most commonly occurs at cysteines within a 5' CpG 3' dinucleotide. Recent research demonstrated that 99.98% of DNA methylation occurs in CpG dinucleotides in the primary human fibroblast cell line, while similar analysis of a human embryonic stem cell line showed that 25% of all cytosine methylation occurs at non-CpG sites. These high levels of non-CpG methylation in embryonic stem cells in contrast to methylation patterns in somatic cells suggest the role of non-CpG methylation is a new active area of research. The frequency of CpG dinucleotides is lower than would be expected throughout most mammalian genomes. However, certain regions of the genome have approximately a 10-fold higher frequency of the CpG dinucleotide than the rest of the genome. These regions are referred to as CpG islands and are often correlated with the location of genes, particularly promoter regions and other regulator regions. These islands compose less than 1% of genome and are typically unmethylated. Another functionally important region for DNA methylation are CpG island shores, which are CpG-containing regions that are within 2 kilobases of a CpG island, contain low densities of CpG sites, and have been shown a location of tissue-specific differential methylation in normal tissues. Methylation of CpG island shores has been correlated with gene expression, and certain shores have shown altered methylation patterns in cancer. Methylation of CpG islands, especially those islands colocalized with promoters or other regulatory regions, is generally associated with gene expression. The mechanisms by which DNA methylation silences transcription are not fully understood, although several mechanisms have been proposed. methylation of a specific regulatory DNA sequence and the attendant insertion of a methyl group into the major group of DNA may prevent stable binding of a regulatory protein, for example, transcription factor, to that sequence, thereby directly preventing DNA activation by a transcription factor. However, many transcription factors are not sensitive to DNA methylation, and not all DNA sequences bound by a sequence-specific transcription factor will contain a CpG dinucleotide. Therefore, other more indirect mechanisms must also be involved in repression transcription by DNA methylation. One such indirect mechanism is what DNA binding proteins containing methylated DNA binding domains. These methylated DNA binding proteins recognize and bind to one or more methylated CpG dinucleotides, which in turn interact with or recruit transcriptional silencing complexes containing chromatin remodeling complexes or histone modification enzymes that act to form a transcriptionally responsive condensed chromatin structure at the associated gene. DNMTs have also been found as components of multi-subunit chromatin modifying and remodeling complexes involved in transcriptional repression. In contrast, intragenetic DNA methylation of active genes is often higher than in promoter regions, suggesting that methylation in the body of genes is a positive regulator of gene expression for dynamic stability. A recent study suggests that this may occur by suppressing the repressive efforts of the polycomb complex, which is associated with chromatin remodeling. 5-hydroxymethylcysteine, the abbreviation is 5-HMC, is one of the hottest topics in epigenetics. 5-HMC is the first oxidative product in the active demethylation of 5-MC. The 3-10-11 translocation TET enzymes oxidize each step in the demethylation of 5-MC, which is first converted to 5-HMC and reactive the gene expression. This process of activation by active demethylation appears to be particularly important for genes involved in brain function, which suggests 5-HMC may greatly deepen our understanding of epigenetics of the brain and development. Mammals have three types of DNMT: DNMT1, DNMT3A, and DNMT3B. DNMT3A and DNMT3B function as de novo methyl transferase to methylate fully unmethylated CpG sites. They function primarily during mammalian development to establish DNA methylation patterns as epigenetic remodeling and reprogramming proceed during differentiation. In contrast, DNMT1 is the most abundant DNMT in cells and is believed to act as the primary mutagens methyltransferase to methylate hemimethylated DNA after DNA replication and preserve path-warp DNA methylation patterns in daughter cells. The fundamental unit of the chromatin polymer is the nucleosome, which repeats every 160 to 240 BP across the genome. Each nucleosome contains a nucleosome core, composed of an octameric complex of the core histone proteins, including H2A, H2B, H3, and H4, which forms a spool to wrap 145 to 147 BP of DNA. The nucleosome core is connected to the adjacent nucleosome core through a segment of linker DNA, which often associates with the linker histone protein H1 or H5. A histone modification is a covalent post-translocational modification to histone proteins, which include acylation, methylation, phosphorylation, ubiquitination, and thymolylation. They can impact gene expression by altering chromatin structure or recruiting histone modifiers. Histone acetylation occurs by the enzymatic addition of an acyl group from acyl coenzyme A. The modifying enzymes involved in it are called histone acetyltransferase, HATs, and they play a critical role in controlling histone H3 and H4 acetylation for open chromatin and gene transcription. More than 20 HATs have been identified, which can be classified into 5 families: GN81, MYST, TAF2250, P200CBP, and nuclear receptor co-activators such as ACTR. Histone deacetylases, HDACs, catalyze the hydrolytic removal of acyl groups from histone lysine residues, which result in the closed chromatin. So far, at least 4 classes of HDACs have been identified. Class 1 HDACs include 1, 2, 3, and 8. Class 2 HDACs are comprised of 4, 5, 6, 7, 9, and 10. Class 3 enzymes require NAD-positive cofactors and include SERTs 1 to 7. The Class 4 enzyme, which contains only HDAC11, has features of both Class 1 and 2. Histone methylation is defined as the transfer of 1, 2, or 3 methyl groups from SAM to lysine or arginine residues of histone proteins by histone methyltransferases. Histone demethylation is defined as the removal of methyl groups from the histone proteins by histone demethylases. In the cell nucleus, when histone methylation changes, specific genes within the DNA complex with the histone may be activated or silenced. Several different histone methyltransferases and demethylases exist that are specific for the lysine or arginine residue which they modify, such as MAL and HST for methylation, while LSD1, Jared1B, and Jared1A for demethylation on histone H3 lysine 4, H3K4. EZH2 for methylation, while UTX and JMJD3 for demethylation on H3K27. Histone phosphorylation and dephosphorylation by kinases and phosphatases has emerged as an important mechanism for the regulation of device events involved in chromatin, including transcription, mitosis, DNA damage repair, and apoptosis. Specific residues in many histones are subject to phosphorylation at specific times and chromosome locations and at particular stages in cell cycle. H2A and H2B are two of the most abundant ubiquitinated proteins in the nucleus. The domain form of the ubiquitinated histones are monoubiquitinated H2A and H2B. A single molecule of ubiquitin is added to the highly converged lysine residues. K119 for H2A and K120 for H2B. Besides monoubiquitination, histone H2A and H2B can be modified by ubiquitinating chains. K6-3-linked polyubiquitination of H2A is usually induced by DNA damage and is required for DNA repair response. Formation of K4-8-linked ubiquitin chains on histone targets them for proteasome-mediated degradation. In addition to H2A and H2B, core histones H3, H4, and linker histone H1 have also been reported to be modified by ubiquitin, but the biological function of these modifications has not been well elucidated. Histone ubiquitination is a reversible modification which are determined by the availability of free ubiquitin and enzymatic activities including enzyme 1, enzyme 2, and enzyme 3. Removing the ubiquitin moiety, on the other hand, is achieved through the action of deubiquitinating enzyme DUB. That's all for this section. Thank you.